Hey everyone, and welcome back to the channel. Today, we're diving into something truly fascinating, something that sounds like science fiction, but is surprisingly grounded in real scientific principles. Imagine this. What if we could extract powerful compounds, even toxins, from plants, without ever cutting them, crushing them, or doing them any harm at all? Just a gentle, almost invisible process. Sounds impossible, right? Well, today, we're exploring a hypothetical concept that might just make it a reality. Before we go any further, a very important disclaimer. The concept we're discussing today is purely hypothetical and for scientific research and educational study only. Please, do not attempt to replicate any of this at home, especially with poisonous plants. Plant toxins are incredibly dangerous and require specialized knowledge and safety protocols. This is a thought experiment, not a DIY guide. So, why is this even an issue? Well, plants are nature's chemists. They produce an incredible array of compounds, many of which are vital for medicine, but some are potent toxins. Think about a plant like foxglove, the source of cardiac glycosides used in heart medication, or the beautiful, but deadly, castor bean. Traditionally, extracting these compounds involves harvesting, crushing, using solvents, or high-heat processes. These methods are effective, but they destroy the plant. They can be energy-intensive, and sometimes the very process can degrade the compounds we're trying to get. But what if we could just ask the plant to give us its secrets gently without causing it any stress? That's where our hypothetical device comes in. We're calling this concept the thermohygroscopic extractor, and the core idea is deceptively simple. We're going to make the plant sweat out its toxins, collect that sweat, and leave the plant perfectly intact. Let's break down the two main scientific principles at play here: humidity and heat. First, humidity. You know how a humid day feels stifling. Plants experience something similar. They naturally release water vapor into the air through tiny pores called stomata. This process is called transpiration, but if the air around them is already saturated with moisture, transpiration slows way down. This builds up internal pressure within the plant. Second, gentle, localized heat. Not enough to cook the plant, but just enough to encourage the water molecules inside to get excited and increase their vapor pressure, especially near the surface. So, here's how our hypothetical extractor would work. Imagine a small, transparent, bell jar-like chamber placed gently over a leaf or a small stem. This chamber isn't just any old dome. It's a precisely controlled micro-environment. Inside this chamber, we first introduce high humidity. This tells the plant's stomata, hey, chill out, no need to transpire so much, so the plant holds on to its water more. Next, we apply very gentle, localized heat to the plant section inside the chamber. We're talking warmth, not scorching. This warmth causes the water molecules within the plant tissue, along with any dissolved compounds like our target toxins, to increase their vapor pressure. Essentially, they want to become a gas. Because the external humidity is so high and we've created this internal push, the plant is gently encouraged to release this warmed, toxin-laden water vapor into the chamber. It's like a focused, low-temperature sweat or distillation. Now for the clever part, the top or inner walls of our chamber are kept significantly cooler, well below the dew point of the air inside. As the warm, toxin-filled vapor rises and touches these cool surfaces, guess what happens? Condensation. It turns back into liquid droplets. These pure toxin-rich droplets then drip down into a tiny collection trough, completely separate from the plant. And the plant? It's perfectly fine, still photosynthesizing, still doing its plant thing. But how do we know the toxins will even come out with the water? This brings us to how plants manage their compounds. Most plant toxins, like alkaloids, glycosides, or terpenes, are either dissolved directly in the plant's water, its sap, or stored in tiny internal sacs called vacuoles. The plant's vascular system, its xylem and phloem, constantly moves water and dissolves substances around. By creating this gentle thermal and humidity gradient, we're essentially encouraging a very localized, non-destructive form of extraction, where the water acts as the perfect carrier for these dissolved toxins. So, if this hypothetical device ever became a reality, what would be the benefits? Firstly, non-destructive extraction. We could harvest compounds from rare or protected plants without harming them. Imagine getting life-saving drugs from a species on the brink of extinction, all while letting it thrive. Secondly, purity. Because we're essentially getting a distilled product, the initial extract might be much purer, requiring less downstream processing. This saves time, energy, and resources. Thirdly, continuous harvesting. Conceivably, you could extract compounds from the same plant multiple times over its lifespan, turning a single plant into a continuous living pharmacy. And beyond just toxins, think about other valuable plant compounds, fragrances, natural dyes, even flavors. The possibilities for sustainable extraction are enormous. Of course, this is a hypothetical concept, and there would be significant challenges to overcome in reality. Things like, how do you perfectly control the microclimate inside that small chamber? How do you ensure the heat is gentle enough not to stress the plant, but effective enough to release the compounds? How do you scale this up from a single leaf to a commercially viable process? And crucially, the selectivity. While we assume toxins would be carried in the water, some compounds might be too large, too non-volatile, or too tightly bound within the plant cells to be released by this method. Further research would be needed to determine which compounds are amenable to this process. But these are the exciting questions that push scientific boundaries. The idea of a thermohygroscopic extractor shows us that sometimes the most elegant solutions come from understanding nature's subtle processes. By playing with humidity, heat, and the plant's own biology, we might unlock a completely new era of sustainable phytoextraction. What do you think of this hypothetical concept? Let me know in the comments below. Have you heard of similar non-destructive extraction methods? We love hearing your thoughts and ideas. If you enjoyed this deep dive into speculative science, please hit that like button, subscribe for more content, and ring the notification bell so you don't miss our next video.